Yeah, thank you for that introduction, and thank you to Mission Bio for having these sessions. I've actually joined one of the previous ones as a viewer, and I think they're a great way for us to all kind of get together and talk about this exciting science during these pandemic times when we're not able to have as many in-person meetings. So I think from my title slide here, which was nicely introduced, you can see basically the overview of what I'll talk about today was when you look at bulk DNA sequencing of splicing factor mutations, they tend to be not co-occurring, or they are actually mutually exclusive. However, there's some rare patients that do have more than one. We termed dual or double splicing factor mutant patients. So we used single cell sequencing with the Mission Bio Tapestri platform to figure out how these cells were able to actually escape from this mutual exclusivity in order to have two splicing factor mutations occurring in the same cell. So just in background to splicing factor mutations in cancer, this is something that's I've studied in the Omar Abdel-Wahab lab at Memorial Sloan Kettering Cancer Center for many years, and these were actually kind of identified in about a decade ago in 2011, occurring mainly in myeloid malignancies, so arising from bone marrow. However, they've also been identified in CLL, which is a lymphoid malignancy, recently in medulloblastoma, which affects the brain, and in a number of other solid tumors, although less frequently. Probably the most frequently is a type of melanoma called uveal melanoma, which is affecting the eye. But today we're going to focus on the myeloid malignancies shown at the top, where mutations occurring in these forms of leukemia or myeloid neoplasms tend to occur in 4 main genes, and these occur almost entirely as heterozygous point mutations as specific residues. So I'm showing at the top U2AF35, also called U2AF1, where you have these 2 hotspots, S34 and Q157. In the middle is SRSF2, where there's a predominant hotspot of P95 mutations. And then finally, an SF34 gene. The hotspots are clustered around this domain right in the middle of the gene. And the most frequent is K700, but there's also others in the 600s, all clustered around that region. The one gene that doesn't fit this pattern is ZRSR2. This is occurring on the X chromosome. And here, the mutations tend to occur throughout the gene and not in a hotspot. And these are predicted to cause loss of function of ZRSR2. ZRSR2 is also unique in that it occurs in the minor spliceosome and not the major spliceosome. However, these mutations do still occur in a mutually exclusive manner with one another, and that has been published in several papers, several observations. For example, in this cancer cell paper from our group in 2018, we did also find the mutual exclusivity of all these genes. These 4 main genes that I just mentioned. However, there's, us and others have noticed there's a few patients here in the overlap region that they have more than one spliceosomal mutation. So even though at a population level, this is occurring mutually exclusive from one another, in these rare individual patients, they have, they seem to defy that epistasis. So the goal of this project was to perform detailed genetic and functional analysis of patients that had myeloid malignancies with 2 simultaneous mutations in RNA splicing factors. And one of the major questions that was only enabled to be answered by Mission Bio technology was, are these mutations occurring actually in the same cell compared to being mutations that are at low allele frequencies in different clones within the population? And then further to that, if they are occurring in the same cell, what is actually allowing them to escape the mutual exclusivity. And so I just want to recognize here two people that helped in the analysis of this data, Xiaoli Mi and Alex Benson, and others that have contributed into amassing this great amount of patient data to find these rare patients, and then analyzing all the sequencing information, including the single cell sequencing information. So we put together this cohort from published data, as well as an additional 1,000 patients plus from Memorial Sloan Kettering that had been unpublished, to get a total of 4,231 patients with myeloid malignancies, including all those listed up on top: MDS, AML, and MPN, and some overlap patients. And you can see that the spliceosomal mutations in these 4 genes account for a fair number of these patients. There's a lot of patients that don't bear any of these mutations. And in those that have splicing factor mutations, there's a strong mutual exclusivity here with a very statistically significant number there when you look at the population level. But then if you zoom in, there are some patients, we've clustered them all on the left-hand side just to make it easier to see, that have more than one splicing factor mutation, so they seem to defy this mutual exclusivity. And this accounts for about less than 1% of patients, 0.85. Looking at this quote double splicing mutant cohort, we identified 32 patients, and you can see there's a combination of all the various splicing factor gene mutations seen. And we had some patients that had mutations, 2 distinct mutations in the same gene, such as those shown here. And so again, our main question was if these are mutations occurring in the same cell. Was there specific mutant alleles that are selected for within these double mutant cohort? And then in these patients that have 2 distinct mutations in the same gene, are they on the same allele? And we were able actually to look just at the bulk sequencing data first and calculate a cancer cell fraction, which is the variant allele fraction multiplied by the copy number of the locus where that gene is present. And in about, so this includes all 32 patients, plus some additional double mutant patients from the Mayo Clinic that were contributed, for a total of 58 double mutant patients. And you can see about half of them, slightly over half, had a cancer cell fraction that was greater than 1 for both of the splicing factor mutations, suggesting that even at the bulk sequencing level, these are probably occurring in the same clone, but again, not definitively shown. So we performed direct single cell genomic DNA sequencing with the Mission Bio Tapestri platform. Here we looked at bone marrow mononuclear cells, sequenced almost a total of 100,000 from 11 different patients, with a median of 7,000 cells per patient. And we here had performed a custom panel where we took 36 amplicons targeting 17 genes. We wanted to focus on the splicing factor mutations and make sure that we were covering all of the splicing factor mutations, including some sort of rare mutations that aren't that common amongst patients. And then we also covered genes that were seen in this cohort of patients that were outside of the splicing factor mutations, but were co-occurring with them. And so we got a good coverage, 192x coverage per amplicon per cell. And just as a first sort of sanity check, we wanted to look at the, like a pseudo allele fraction taken from the single cell data and compare that to the bulk sequencing allele fraction. And we saw a good concordance, so that kind of told us that we're getting good data from the single cell sequencing. And then just focusing on the splicing factor mutations and ignoring for the moment the other co-occurring mutations, we looked at these double mutant patients at the 2 splicing factor mutations. So in this example shown here, it's a patient that had U2AF1 Q157R and also a SRSF2 P95H. And so you can see in the gray bars are the single cells where they had only a U2AF1 mutation, and then in the red bars are single cells where there were both mutations present, and we did not see any representation of the SRSF2 by itself. So we can here presume that the U2AF1 mutation occurred first, followed by acquisition of the SRSF2 mutation. And they are indeed in those cells occurring in the same cell. And these are more examples. Actually, a theme that came up was that the SRSF2 mutations tended to occur following the other spliceosome mutation in these patients that we sequenced in these examples here. The one exception was that we never saw SRSF2 P95H and SF3B1 K700E occurring together. So although we might have saw other SF3B1 mutations such as H662 or K66 here, co-occurring with SRSF2 P95, we did not see SF3B1 K700E occurring with P95. So to summarize this a little bit, they do coexist in the same cells. There may be some allele-specific preferences, and notably the P95H and the SRSF2 P95H and SF3B1 K700E specifically did not co-occur, and I'll speak to that in a moment. Here's some of the other samples that we sequenced where you have a U2AF1 and an SF3B1, or ZRSR2 and U2AF1. Another case where we had mutual exclusivity was these 2 ZRSR2 mutations shown at the bottom. ZRSR2 is on the X chromosome, and in this male patient, there's only one copy of ZRSR2, and in fact here we did not see them occurring in the same cells. So these are 2 different clones within that patient having 2 different ZRSR2 mutations. Now here we're adding in so these are the same samples, but adding in the other mutations that are not splicing factor mutations, such as TET2, DNMT3A, SXL1, a lot of other very common mutations seen in myeloid malignancies. And on the far right, you can see we built these fish plots, and notice that typically in these double mutant patients, there was a preceding mutation, such as TET2 in these examples, that preceded the acquisition of any splicing factor mutation, and then the double splicing mutation tended to occur later. And again, we did not see the SF3B1 and the SRSF2 co-occurring, the most common hotspots of those 2 genes. These are some more examples, just showing you the same data. And here's the ZRSR2 mutant patient that had the mutually exclusive mutations in ZRSR2. So we tried to look at other potential causes for escape from mutual exclusivity, such as maybe these are unique disease subtypes. But when you compare the patients with 1 splicing factor mutation to those with 2 splicing factor mutations, really the representation of the diseases is very similar, so there's no difference by disease subtype. We also thought that there might be a potential for just an increased mutation burden in these double mutant patients, But you can see from this plot here, there was no clear difference in the tumor mutation burden in the double versus single mutants. So it's not explained by those causes. We did, however, find that there was a bias in the mutant alleles selected for in the double mutant patients. So I'm showing here the single mutant patients. Remember, this is 4,000 some patients, and the representation of the splicing factor mutant alleles. So again, SRSF2 P95 is the hotspot in that gene. SF3B1 K700E is the most common hotspot in SF3B1, and then we grouped that against all others. And what we noticed was that there was selection against the most common SRSF2 and SF3B1 mutations in the double mutant patients. So whereas those are the most common in the single mutant patients, you'd expect that they would also be very common in the double mutant patients. However, they were less frequent in double mutant patients than in single mutant patients. So that suggested there is an allele-specific bias in these double mutant patients. And we also noticed one patient had a very rare SRSF2 P95 mutation, P95A, which is very uncommonly seen. You typically would get P95H, L, or R, so the exact amino acid substitution can be different. And we tested these various mutations. The P95H, L, and R are known to have a specific effect on binding of SRSF2 to RNA by changing the binding affinity of the SRSF2 protein to RNA. However, you can see at the bottom there, the P95A variant did not change the binding affinity. So essentially this mutation has no effect. It's almost a missense mutation in that terms that it does not affect the binding of SRSF2 protein, as do the more common amino acid substitutions. So in that particular patient, that might be the reason that they were able to have two splicing factor mutations, is that one of them, the SRSF2 P95A, did not actually affect splicing in terms of binding of SRSF2 protein. The other thing we saw that there was a selection for these other SF3B1 mutations that are non-K700E, so these other SF3B1 mutations such as K666 or others. And to look a little bit further into this, we took a cohort of single splicing factor mutant patients that had either the SF3B1 K666 mutation or the SF3B1 K700E mutation. And we performed total RNA sequencing and calculated aberrant splicing seen in those patients. And on the left, we plot the top 20 SF3B1 K700E misspliced genes, and then plotted them. This is the change in the percent spliced in, so it's a measurement of aberrant splicing, and the higher the fraction, the more aberrant splicing. And on the bottom, we have the SF3B1 mutant allele fraction. So you can see as the allele fraction increases for SF3B1 K700E, you see an increase in the aberrant splicing of these top 20 misspliced genes for K700E. And the missplicing is stronger at all of these as compared to SF3B1 K666. Now on the right, when you do the converse, when you take the top 20 K666 misspliced genes, and then you look at the percent spliced in by allele fraction for the SF3B1 K700E or K66, you see that the K700E actually missplices these in a similar strength, if you will, as the K666. So essentially, this is telling us the K666 has a weaker effect on splicing of these target genes. in the K7RE target genes or in the K666 misspliced target genes. So essentially, this may allow the patients with these non-K700 splicing factor mutations in SF34 to have tolerate a second splicing factor mutation, such as in SRSF2, without causing the synthetic lethality that we know occurs when you have too much aberrant splicing. And then another pattern that we noticed was enrichment of two mutations in U2AF1, so having a co-occurring S34F and Q157R. And this is an example of a patient where on the single cell level, essentially all greater than 1,000 cells that we sequenced that had either of these splicing mutations, they were always seen in the same cell, and they were not seen in single cells alone. So essentially, these mutations appear to have occurred at the same time. And using those same samples, we did some subcloning and long-range PCR to look if these mutations occur in cis or if they're on different alleles. And you can see every time that we found a wild type allele in the S34, we always see a wild type in the Q157. And every time we found a mutation in S34, we always found a mutation in Q157, suggesting these are indeed occurring in cis. And we replicated, we had a second patient that unfortunately we didn't have material for the single cell sequencing, but we were able to perform the same cloning in long range PCR. And in both cases, they occurred in cis. We did have bulk RNA sequencing for both of these cases, and we compared that to some of the bulk RNA sequencing from patients with either the S34 alone or the Q157 alone. And you can see here the double mutant patients in the middle have overlapping splicing changes with both of those categories. So this suggests potentially, in the case of U2AF1, having these two mutations in U2AF1 may have some additive effect, that you're getting the benefits in terms of the oncogenic benefits of both of these mutations synergistically, additively occurring together. So just to conclude, and happy to take any questions, these mutations are rare, but it is a clinical question that occurs with, it occurs enough that we and others have observed it and wondered about this. And then in terms of the basic level, I think this also gives us insight to how these are escaping epistasis, and it's potentially through the allele-specific effects on splicing and RNA binding. So I just want to acknowledge Omar Abdel-Wahab's lab, where a lot of this work happened at Memorial Sloan Kettering, Rob Bradley's lab at the Fred Hutchinson Cancer Center, who performed a lot of the analysis in terms of the bulk RNA sequencing, and samples that we got from the Mayo Clinic from Mrinal Patnaik, and Stephanie Helane's lab at Yale, who helped with some of the SRSF2 binding, and to Mission Bio for all their support in the Tapestry experiments. So this was published in Blood. I hope you all can check it out at this link. And please email me or reach out on Twitter with any questions. Thank you.
Thanks so much, Dr. Taylor. So we have a couple of great questions that have come in, but I want to take the first one. I'm just kind of interested. What made you want to study this very rare phenomenon?
Well, we've been studying splicing factor mutations in general in terms of how they might be causing cancer, and then whether they could be targets of drugs for patients with cancer. And it was an observation that we noticed that these happen in these rare patients, but they really shouldn't be happening if you look at the population level where they're mutually exclusive. And we've done some modeling in animals where they also cause synthetic lethality if you see these splicing factor mutations together. So it was very much intriguing, and we'd had some experience with Mission Bio Tapestry in terms of knowing the power of that for single cell sequencing, and we thought it would be a great way to answer this question.
Huh. That actually leads up to another question. It's great that the Tapestry platform can tell whether these mutations are co-occurring in the same cell, but if you didn't have the Tapestry platform, is there an older technology that you would have had to use to answer this question?
Yeah, you can do it. There's a very difficult way to do it is single cell colony assays, where you take some of the bone marrow cells and you can grow them into, you split them up into single cells, and then you can grow them in a special media that makes them into single cell colonies, and then perform sequencing for each of these alleles in those colonies, and you do that for hundreds of colonies so that you can basically re-represent what you're able to do very quickly on the Mission Bio Tapestry platform. And so it saves a lot of time. It's probably even, with the number of cells you're able to do as compared to just counting 100 colonies, you're able to do thousands of cells. So it is probably also more accurate.
I don't want to date you, but did you ever have to do that? And is that what you did with the UTF-1 data?
Yes, yeah. So I've done that, and I've done that actually very recently too. Some reviewers wanted to actually confirm single cell sequencing data. So we've done that just to prove also that, not in this particular example, but in others, that actually single cell sequencing is as good or better than that technique.
Yeah, it's interesting to see Sanger sequencing data, and there's still a place for that when you need a cheap option to screen many, many different clones.
Yes, yes.
Does this happen in any other disease besides oncology?
Splicing factor mutations? No, not that we're aware of, although splicing or aberrant splicing is important for lots of diseases, developmental diseases, or neurological diseases. And there's, lots of literature about aberrant splicing and ways to correct splicing to fix those kind of classical genetic diseases. But these mutations that I mentioned here are only mutated in cancers as far as we know.
Got it. Let's see here. I want to grab a good question. So do you consider the comutation of PRPF40B with SRSF2 also rare?
PRPF40B, I would say, yeah, I would imagine would be exclusively rare. We, again, just focused on these most common splicing factor mutations. So there are, as this question is probably alluding to, other splicing factors that are mutated in these cancers, but they're far less common than these four genes. And so for this particular paper, we only focused on those most common in order to be able to, do some of the statistics in terms of, we didn't study anything that was, only occurring very rarely.
A follow-up there, how many genes did you cover with your DNA panel?
We had 17 genes, and so those were the 4 splicing factor genes, and we actually had multiple amplicons for those. And then in terms of other genes, they were selected for based on the patients that we were sequencing. So we knew the bulk sequencing data for the patients, so we knew which mutations were present already. And then we were able to design the panel based on those genes.
Great. We have a nice comment. So they said, very nice talk. I understand there were technical difficulties sequencing the SRSF2 mutations on Tapestry. Can you comment on these and whether you managed to overcome them? If not, do you think they're surmountable on this platform?
Yeah, thanks for that question. It's a great question. Actually, if you go to the paper, we talk a lot about that and have some experiments in the supplement where we delineate that. So the SRSF2 particular region is very GC rich, and I think that it had a difficult time sequencing. We saw some allele dropout, which is where you have, instead of both alleles being sequenced, you just have one. So these mutations are occurring as heterozygous mutations. So we would like to see one wild type copy and one mutant copy. And in some cases we saw, just one mutant copy without any corresponding wild type copy. However, we know that from bulk sequencing data and lots of data that we've generated in other studies, that these do occur as heterozygous. We don't see them with a loss of heterozygosity or amplification of that locus. And we actually confirmed that with these patients that they only had 2 copies of SRSF2. And so it didn't make sense to just see one mutation. So we tried to model different methods, and that's explained in the paper, to basically look at the co-occurrence, making different presumptions, presuming if there was one mutation there, that was a heterozygous mutation and not homozygous. And for those reasons that I just explained, I think it was a pretty safe assumption. So it's not insurmountable. I think if you can do it the way we did it, I think we've talked to Mission Bio again about maybe ways to increase the performance at that specific gene. But I think that's in the works. For the time being. If you have already generated the data and you wanted to try to analyze it, you can use our methods, and I'm happy if you reach out to me to share that with you.
Great, thank you. Are there drugs that target these RNA splicing factors? Would you change the treatment if you found 1 versus 2 mutations co-occurring versus not? How do you think your findings will inform therapy strategies?
Another great question. There are some drugs in development that inhibit splicing, and they are being developed because we and others had found that in patients with 1 splicing factor mutation, if you give them a drug that inhibits splicing, you can cause them to undergo apoptosis because they reach a tolerance level of the splicing is too perturbed, and they triggers apoptosis because you get a buildup of all these misspliced genes. And so, none of these patients with 2 mutations have been tested with that drug, but we presume, given that they have 2 mutations, that is more missplicing than just 1 mutation, so that they would still be sensitive to these drugs that inhibit splicing by that mechanism. But it's a great question. We haven't been able to test that, and I don't think it would change the sort of idea of the paradigm of being able to inhibit splicing in these splicing factor mutant patients.
Great. Do the mutation of epigenetic factors is frequently co-occurring with the splicing factor mutations? So I think the question is, the mutation epigenetic factors, does it frequently co-occur with the splicing factor mutations?
Yeah, and in these particular patients with the double splicing factor mutations, we saw that all of them, in fact, had a co-occurring and probably occurring first mutation in an epigenetic factor such as TET2, DNMT3A, or ASXL1. We don't yet know exactly if that is required in order to be able to have 2 splicing factor mutations, or if it's just a reflection of how common it is to see these mutations together in general.
Yeah, I was really excited when I read that in your paper, because I think from other Mission Bio Tapestry papers, they've been finding that mutations are occurring in epigenetic factors prior to signaling pathways, and now you found splicing factors, so it very much supports Kathy Smith and Ross Levine's findings.
Absolutely.
All right, we have a lot of questions coming in. Thank you for this great talk. Do you know if splicing factor mutations in U2AF1 or ZRSR2 are by themselves able to start a myeloid neoplasia? Or would they need additional mutations such as the ones you described or in other epigenetic genes?
Yeah, well, so we know from what we can study in mouse models, and when you have U2AF1, for example, is not our work, but others, when they induce single mutation in U2AF1, you see some changes that you would associate with myeloid malignancies, such as change in the blood counts or dysplasia, mild dysplasia in the mice, but not developing the full phenotype. And so that does suggest you likely need some other mutation. And as you referred to these other papers that look at the clonality of acute myeloid leukemia, for example, patients tend to have more than just one mutation, typically 3 to 5 mutations, so driver mutations. So I think combining the mouse modeling data with what we know from patient sequencing, you likely need to have more than just the single splicing factor mutation.
Great. All right, I think one final question for me. I know this work was done in conjunction with a lot of groups and at Memorial Sloan Kettering. Now you're a new assistant professor at University of Miami, and I'm very curious what your lab is going to study. I love your paper and how it followed clinical samples, but also was very heavy in the biochemistry. And so what do you intend your lab to focus on?
Oh yeah, thank you for that. I did just start here at the University of Miami. It's a great place to work and a great group. I've been excited, even though it's challenging with the whole pandemic going on around us. But I'm going to focus on something that I studied in addition to the splicing factor mutations, which is nuclear export and this gene called XPO1, which is also mutated in these types of cancers. And we actually have an interesting finding recently too, that there's drugs that target this nuclear export pathway, and those have had some effect on splicing in these patients that we studied with MDS specifically. So I'm digging into that right now to figure out what's the connection between nuclear export and splicing, and basically how we can target these two pathways in patients to hopefully improve outcomes of these diseases.
Great. Well, I just want to thank you for a fantastic talk. Everyone, please check out the paper in Blood. And thanks again, Justin. Good luck starting your new lab, and I hope you get through all this COVID era.
Thank you. Same to you. Stay safe.